Good morning friends. Today we learn about paper number 3 from MSc second year Research Methodology, Insect Physiology and Biochemistry. Topic circulatory system of insects, that is components of circulatory system. In insect, circulatory system is open type of circulatory system. In animal kingdom, two types of circulatory systems are present. First one is closed type of circulatory system and second is open type of circulatory system. Before starting components of circulatory system, we learn about closed circulatory system and open circulatory system. In closed circulatory system, that is closed vessels are present, blood flows into closed vessels, that is arteries, veins, capillaries. And in open type of circulatory system, blood flows directly into the body cavity, that is sinuses. And these closed circulatory system present in higher vertebrates and open circulatory system opens in lower invertebrates. This closed type of circulatory system mainly carries nutrients, gases as well as excretory products, but in open type of circulatory system only nutrients and excretory products are carried. This open type of circulatory system present in insects. In insect, body cavity is called hemocoel. Body cavity is called hemocoel and blood present in insect, it is called as hemolymph. Blood present in insect is called hemolymph. This hemolymph is circulating fluid or blood of insects. Insects hemolymph differs from vertebrate blood with absence of erythrocytes, that is RBCs are absent in the insect blood or hemolymph. Again, high concentration of amino acids are present in hemolymph. This hemolymph color is white in color or colorless fluid. Sometimes it is greenish yellowish fluid containing large number of varying shape of hemocytes are present. This hemolymph is colorless and in hemolymph varying size hemocytes are present. pH, hemolymph pH is slightly acidic in pH, range between 6 to 7. pH is present in insect hemolymph and slightly acidic in nature. Specific gravity of hemolymph, is 1.01 to 1.06 specific gravity is present. This specific gravity depends upon sex and developmental stages. Next point is chemical composition of hemolymph. These hemolymph mainly consist two components. First one is plasma and second is hemocytes. The composition of hemolymph in insects varies depending upon the diet, temperature, stages of development. It is mainly composed of two components. First one is plasma and second component is hemocytes. Plasma. This plasma contains about 85 to 90% water is present in plasma. This plasma is clear but sometimes pigmented with the help of color pigments, that is yellow, brown, green, orange and red pigments are present. Green color may be present due to chlorophyll is dissolved in the plasma, and red color due to presence of hemoglobin in the hemolymph. This plasma also contains high concentration of amino acids, proteins, sugars and inorganic ions. If look at the ionic composition of hemolymph, this hemolymph shows rich in potassium ions in phytophagous insects and sodium ions in predator insects, rich in sodium ions. Again, this hemolymph contains hormones, and these hormones are diuretic hormones, uretic hormones, and these hormones regulate the water balance in the body of insects. Some insects consist pigment cells, that is xanthophyll pigments is present, as well as hemoglobin is present in the hemolymph of insects. Next point is hemocytes. These hemocytes are first discovered by scientist Arnold in 1974. These hemocytes first discovered by Arnold in 1974. These hemocytes are blood cells that circulates in the hemolymph. These hemocytes are amoeboid, nucleated nucleus are present. They are homologous with vertebrate leukocytes. These hemocytes are homologous to vertebrate leukocytes, that is are WBCs, similar to the WBCs, and circulates freely in the hemocoel. In most insects and larvae, the hemocytes are produced within the hemopoietic organs. These hemocytes are produced in the hemopoietic tissues. Hemopoietic tissue is produced different types of hemocytes, and these hemocytes are several types and nucleated. These hemocytes are different types. First type is prohemocyte. It is also called as proleukocytes. These hemocytes are smallest of all cells, and nucleus is spherical in shape with large nucleus present. Again, granular when stained, these are basophilic in stain, and these prohemocyte shows mitotic division and generate new other cells are produced from prohemocytes. Next type is plasmatocytes. It is also called as amoebocytes, and these cells are abundant, which comprises more than 30% of total hemocytes. These plasmatocytes involved in phagocytosis. Phagocytosis means cell use plasma membrane to engulf foreign particles, that is viruses, bacteria and insect, protect from foreign particles. These cells again involved in encapsulation, and this encapsulation defense mechanism acts on metazoa parasites. Then surrounding the parasites, several layers are produced. These layers are cemented with the help of intercellular cells, and this encapsulation leads to death of parasites, and this again protective mechanism for the defense. Next type is granulocytes, granular hemocytes. These granular hemocytes are oval in shape, small nucleus and large cytoplasm is present, and this cytoplasm having granules, non-motile but metabolic in function. These granulocytes involved in metabolic functions. Next type is adipocytes. It is also called as adipocytes, adipohemocytes. It is also called as lipocytes. These adipocytes are large oval cells having eccentric nucleus. Eccentric nucleus means this nucleus is shifted from the center. Nucleus is not presented in the center. It is called as eccentric nucleus, and eccentric nucleus present in adipocyte cells. These adipocyte cells cytoplasm shows distinct fat droplets are observed in the cytoplasm. Next type is coagulocyte. It is also called as cystocytes. These coagulocyte cells are spherical cells with large nucleus and involved in coagulation, that is blood clotting mechanism. Next type is podocyte cells. These podocyte cells is also called as stellate cells, and these stellate cells having large nucleus and number of protoplasmic projection are observed. Look at this diagram. This diagram shows protoplasmic projections. And last type is vermiform type. These vermiform hemocytes thread-like spindle-shaped structure with granular basophilic cytoplasm is present, and these are rarely occur, mostly occur in the larvae. Next step is spirule step. This also called as spirulocytes. This not very common but involves in the secondary function. These are different types of hemocytes. These hemocytes involved in different functions. Hemocytes perform different functions. First function is lubrication of the tissue. Hemolymph performs different functions. Hemolymph function. Hemolymph functions. First function is lubrication. Lubrication. This hemolymph keeps the internal cell moist and movement of internal organ is also made easy. Second function, transport and storage. Transport and storage. These hemolymph digested nutrients, hormones are transported with the help of hemolymph. It also removes the waste material to the excretory organs and each hemolymphs, water and raw materials required for histogenesis is stored in the hemolymph. Third function is protection. Hemolymph protects. It helps in phagocytosis. This phagocytosis is again cell use plasma membrane to engulf foreign particles, as well as encapsulation, detoxification. Toxic substances are removed from the insect body. Coagulation, that is blood coagulation, and wound healing. In site of wound healing, connective tissue formed and wound is held. Non-cellular components like lysosomes also kill the invading bacteria. These different mechanisms used to defend the insects, that is phagocytosis, encapsulation, detoxification, coagulation and wound healing. Next is heat transport. Heat transport. In this heat transport, generally heat is hemolymph movement in the circulating system, regulate the body heat. It is called as thermoregulation. Body heat is maintained through the circulating fluid. It is called as thermoregulation. Next, maintenance of osmotic pressure. Next function is maintenance of osmotic pressure. Maintenance of osmotic pressure. In these ions, amino acids, organic acids present in the hemolymph helps maintaining osmotic pressure required for normal physiological function. Next point is metabolism. These hemocytes are involved in for ongoing metabolic reaction, that is Trehalose present in the owl insect. In owl insect, trehalose sugar is present, and this trehalose sugar is converted into glucose. Next point is immunity. Immunity. This immunity term used for the ability of an organism to resist disease. While in insect, immunity is used to express defense reaction of insects against the infection. Immunity used to express defense reaction of insects against infections. And last point is connective tissue formation. Connective tissue formation. These hemocytes are involved in connective tissue formation, that is, membranous sheath is produced in site of wound healing, and membranous sheath is produced, that is, hemocytes are involved in connective tissues. Thank you.